This lecture is on skeletal muscle, structure, and function. This is the overall outline of Chapter 8. Skeletal muscles are very important for your overall health. They make up about 40 to 50% of the total body weight. They have various functions. There's force production for locomotion and for breathing. Skeletal muscles are voluntary. So any kind of muscle where you can voluntarily control would be skeletal muscle. Force production for postural support. Skeletal muscles are used for heat production during cold, so for shivering. And then finally, skeletal muscles are also used as secretory gland organs. So they can secrete cytokines and other peptides, and these can exert an autocrine, paracrine, or endocrine effects. So this is kind of new research, but it's now shown that skeletal muscle do have this interesting extra function that people hadn't really known about earlier. Muscles have two different actions. They can be flexors, meaning that they decrease the joint angle. For example, the biceps decrease the joint angle at the elbow. The triceps are extensors. They increase the joint angle at the elbow. So muscles are organs. They consist of all four basic tissue types, which would be connective tissue, neural tissue, muscular tissue, and epithelial tissue. So skeletal muscle, and we'll go through explaining that in a second. Skeletal muscle, as its name implies, generally will attach to muscle via tendons. There are a few exceptions to this rule. There are some skeletal muscles that do not connect to bone. Skeletal muscle is generally under voluntary control. In other words, with smooth muscle and cardiac muscle, you can't voluntarily control them. They're contraction, but with skeletal muscle, almost all skeletal muscle, you can control them voluntarily. So let's look at this example of a skeletal muscle. Here we have connective tissue, so the tendon would be considered connective tissue. We have fascia, which is connective tissue. So surrounding the muscle itself would be epimysium, which is a layer of connective tissue. Now, the muscle, within a given muscle, the muscle fibers are organized into fascicles. A fascicle is a bundle. And around each bundle is perimysium, which is also connective tissue. Now, if we take a fascicle and look at it in more detail, we'll see that within the fascicle, there are a number of muscle fibers. A muscle fiber is a muscle cell, so it's a single cell. And so you can see there's a number of muscle fibers within a fascicle, and there's yet more connective tissue. It's called endomysium between the individual muscle fibers. Now, if we're to pull out an individual muscle fiber, as shown in this diagram, you can see that within it, there are a number of rod-like contractile units, which are called myofibrils. So these are the structures made out of protein that will actually do the contraction, the work of contracting. And as we'll see, that the myofibrils are largely made up of two types of filaments, thick and thin filaments. And we'll talk more about those in a little bit later. Now, a single muscle fiber in a skeletal muscle is multinucleate, meaning it has multiple nuclei. And so it's hard to see in this picture, but it's supposed to indicate here that there are multiple nuclei within a single cell. Now, these actually start out as developmentally, they start out as individual separate cells, each with its own nucleus, but then they fuse together to form this much longer cell. In this picture, we can also see a satellite cell. We'll talk more about that in a second. This nucleus is separate. It's a different nucleus. It's not the nucleus of the actual skeletal muscle fiber. This is an adult stem cell. So it's a cell that's able to continue to divide and differentiate. We'll talk more about that in a second. So far we've shown that there's connective tissue, there's muscle tissue. Now there's also circulatory system with vessels. So vascular tissue within the muscle itself. So you can see here, there's an artery, and here's a vein shown in blue. And so those have epithelial layer, right? So that is epithelial tissue. It's called endothelium when it's in the cardiovascular system. And then finally, you can see that this muscle is innervated, meaning that a nerve comes to it and makes contact with it. And we'll talk more about that in a second. So those are all four different four basic tissue types within a single muscle, which would then therefore be considered an organ. Okay, so I think that's it. So we'll move on to the next slide. Okay, this slide is largely a recap of some of the things we just talked about. So there were in terms of connective tissue, we have the epimysium, which surrounds the entire muscle, the perimysium, which surrounds bundles of muscle fibers or fascicles. We have the endomysium, which surrounds the individual muscle fibers. Now, there's also a basement membrane. This is a thin layer of fibrous extracellular matrix. It lies just below the endomysium, and it's very important for things like muscle regeneration. And the basement membrane itself is composed of two layers, the basal lamina and the reticular lamina. The basal lamina is directly linked to the sarcoplasma, so it's the closest layer into the sarcolemma. And the sarcolemma is just a different way of saying plasmalemma. So normally a cell's membrane is called the plasmalemma. For a muscle, it's called the sarcolemma. And the reason for this is because sarco, which means flesh, fleshier of the flesh, comes from Latinized form of Greek sark. So this is, instead of plasmalemma, it's called sarcolemma. I mentioned satellite cells before. These are very important. They play a key role in muscle growth and repair. So during muscle growth, satellite cells can increase the number of nuclei and mature muscle fibers. So what they do is, you see how they lie very close to the muscle itself. And if need be, they can divide. So they'll retain one nucleus as an adult stem cell. And then the second one will differentiate and it'll enter into the muscle fiber, and it'll become a new nucleus. And so this is necessary because each nucleus essentially governs a certain domain called the myonuclear domain. So this myonuclear domain is the volume of cytoplasm surrounding each nucleus. And so it directs essentially protein synthesis and controls a particular area or domain of a muscle fiber. So for example, if you exercise, your muscle fibers will get larger in volume. And so there should be an increase in the number of corresponding increase in the number of nuclei. And that's exactly what happens when you exercise. These satellite cells will divide, and there'll be an increase in the number of nuclei. So this increase in nuclei allows for greater protein synthesis, which would be necessary. Remember, these muscle fibers are largely protein. They have all the other things you'd expect in a cell. They have mitochondria, they have all the other organelles, lysosomes, things like that. But they're also really packed with protein, these thick and thin filaments, which are within the myofibrils, which are necessary for contraction. So there's a tremendous amount of protein synthesis going on all the time. And finally, the satellite cells are important for muscle growth in response to strength training. In this slide, we've zoomed in on a myofibril. So this is what you can see here is a single skeletal muscle fiber. It'll have, again, it's multinucleate. It has associated satellite cells. And within the muscle fiber, if you do a cross-section, you'll see there are a number of myofibrils. And so we're pulling one out here to look at it. And within the myofibril, the actual unit that does contraction is called the sarcomere, and that's going to contain both thick and thin filaments. The thick filaments are shown in purple, and the thin filaments are shown in sort of a greenish blue here. But the sarcomere is delimited by what are called the Z lines, or sometimes it's referred to as a Z disc, and these are made out of proteins. And so each sarcomere itself is capable of contracting or reducing its length. So if we look, for example, at one sarcomere, you'll see that the sarcomere starts from one Z line and it ends at the next Z line. And then there are a number of different bands and lines in there. There's, for example, the M line, and M essentially is the middle line. There will be a middle line due to a lot of protein being at the very center. And this protein is associated with the thick filaments, which, as I said, are shown with this sort of purplish color. And then associated with the Z discs, it's hard to see here, but there are thin filaments, which are shown here in this bluish green here. You can't really see in this figure here. Well, actually, if you go down here, you can see them here. So here are the thin filaments. Okay, and you can see there's a lot of overlap between the thick and the thin filaments. And if we actually want to look at or understand the proteins that are involved, the thick filaments are largely made up of a protein called myosin, and the thin filaments are mostly made up of a protein called actin. And we'll come back and talk to this topic and talk more about how the contraction actually occurs. Here's a three-dimensional picture of or diagram of skeletal muscle fiber. So you note that within the muscle fiber, there are multiple myofibrils. And each myofibril starts from one Z disc and ends at another Z disc, probably around here. You'll again note that this muscle fiber is multinucleate. Now I want to point out something else. So this muscle fiber, of course, has the sarcolemma, right? The plasma lemma of the fiber itself. This plasma lemma actually has these invaginations that go all the way through from one side to the others. And these are called transverse tubules. In other words, let me draw a picture of this. If you had, if you were looking at the surface of the cell, it actually dives down and into the cell. And in fact, they're called transverse tubules. They actually go all the way through and exit out the other side of the muscle fiber. And so these are really important. As we'll see later, what happens is there will be a wave of excitation, and that's transmitted along the surface of the cell. It's a change in voltage across the cell, and this will travel along. And if we didn't have these transverse tubules, this wave of excitation would simply pass all the way from where it started to the very ends of the muscle fiber. But the issue is that we need to couple this excitation of the muscle fiber. It's an electrical excitation with the actual contractions. This is referred to as excitation-contraction coupling. And in order to do that very quickly, we need to bring the electrical signal into the neuron, into the muscle cell. And so that's what these transverse tubules are for. They're essentially continuations of the plasma membrane that dive into the cell. And this allows for this electrical signal to pass very quickly throughout the entire cell, including deep down within the cell. Now, associated with the T-tubule are the terminal cisternae. So let's draw that. Not a very good drawing. So within a muscle cell, you have endoplasmic reticulum, but instead of being called endoplasmic reticulum, it's called sarcoplasmic reticulum. And near each T-tubule, there are these things called terminal cisternae, which are shown here in blue. You see how it's sandwiching the T-tubule, which is shown in purple. And so the sarcoplasmic reticulum stores calcium, and this calcium will be an important signal that leads to contraction of the muscle fiber. So it's important to understand this three-dimensional structure here, that you have the sarcolemma, which is the plasmalemma. In order to bring the electrical signal, you have these continuations of the membrane into the cell. And those are called transverse tubules, or sometimes just called T-tubules for short. And then associated with that are these terminal cisternae of the sarcoplasmic reticulum. And their primary function is to store calcium. And they will release calcium, and that calcium will serve as a signal for muscle contraction. Another important anatomical structure is the neuromuscular junction. Motor neurons, called somatic motor neurons, will have their cell bodies in the spinal cord. They'll exit through a spinal nerve together with other axons, and they'll travel out to the periphery where you have the skeletal muscle. And on each skeletal muscle, they'll make a number of connections. And these connections are called neuromuscular junctions. So for example, here is an electron micrograph, or maybe it's a light, it might be a light level microscopic image, but I just drew on this. But you can see here, there are a number of these structures, and those are neuromuscular junctions, and these lines are axons. So now if we zoom in this diagram, we can see here, we have a motor axon coming in. It's myelinated, meaning it has a layer of insulation on it. At this structure here called an axon terminal, or sometimes it's referred to as a synaptic bouton. Bouton means button in French. There is neurotransmitter within these, and this neurotransmitter will be released into what's called a synaptic cleft, and it will diffuse across, and there will be receptors, protein receptors, in what's called the end plate. Okay, so at a neuromuscular junction, there's a specialized bit of membrane right at the neuromuscular junction called an end plate. And you'll note that the end plate has some characteristics. It has infoldings, so this increases its surface area. And it also has a lot of protein, which is shown here in blue. These are receptors for the neurotransmitter. So the neurotransmitter diffuses across a short gap. It binds with these receptor proteins, and then this will initiate an effect in the postsynaptic cell, which is the muscle cell here in this case. The neurotransmitter of interest here is acetylcholine. It's released from the motor neuron, and it causes what's called an end plate potential. So normally the muscle cell has what's called a resting membrane potential, meaning it has a negative charge with respect to outside of the cell. When the acetylcholine is released and interacts with those receptors, you'll have a phenomenon called a depolarization, in which the membrane becomes less polarized, in other words, less negative. And this can lead then to the activation of what's called an action potential within the muscle fiber. A single motor neuron that innervates skeletal muscle will typically innervate more than one muscle fiber. In fact, it may innervate hundreds of muscle fibers. So if you look within a muscle, in this case, we have a muscle with just, I don't know, about a dozen muscle fibers, just for illustration purposes, and we have just 3 motor neurons. Now, normally the numbers, of course, would be much higher than this. But you can see that here that each motor neuron will go to a subset of the motor neurons. So for example, the motor neuron with the blue cell body here has an axon that goes out to the periphery. It innervates this skeletal muscle, and we can see that anything that's blue is a blue muscle fiber is being innervated by the blue motor neuron. There's sort of a purple one. So this purple motor neuron will innervate certain subset of the muscle fibers, which are indicated in purple. And then finally, we have sort of a pinkish red motor neuron, and it goes to another subset. So an individual muscle fiber is only innervated by a single motor neuron, but a single motor neuron innervates multiple muscle fibers. And the reason for having, the combination of a motor neuron and all of its postsynaptic muscle fibers is called a motor unit, and they work together. In other words, when an action potential, which is a nerve impulse, travels along the axon of the motor neuron and it reaches all these synapses or motor neuromuscular junctions, it will activate each of these muscle fibers simultaneously. And the reason for having this kind of design is that sometimes you don't need the full strength of a muscle. It wouldn't make any sense if you're just picking up a coffee cup to use your full strength. And so you want to use a subset of the muscle fibers. And so you would just activate a certain number of motor units and not all of them. Here are a few take-home messages about the things we've just discussed. Skeletal muscle constitutes about 40 to 50% of the total body weight. Skeletal muscle performs three major functions: force production for locomotion and breathing, force production for postural support, and of course, skeletal muscle used for heat production during cold stress, in other words, shivering. And fourth, we know now that skeletal muscle also has a secretory function. It can generate autocrine signals that are released from the muscle but have an effect back on itself. They can release paracrine signaling molecules that act in the local area. And they can also release endocrine signaling molecules. These are ones that actually go throughout the bloodstream and act as a hormone to have effects on other organs. And that's relatively recent research, but it's very exciting and interesting, so it's worth bringing up. Individual muscle fibers are composed of hundreds of thread-like protein filaments called myofibrils, and we'll talk more about that a little bit later. Myofibrils contain two major types of contractile protein: actin, which is part of the thin filament, and myosin, which is a major component of the thick filaments. One characteristic of skeletal muscle is that they're multinucleate. They start out developmentally as individual cells, but then they fuse together. And so each nucleus has what's called an area of cytoplasm, which is called its myonuclear domain. So it's essentially in charge of protein synthesis and so forth within that region. And so the importance of this myonuclear domain is that a single nucleus is responsible for gene expression in just that surrounding region of cytoplasm. So as you can imagine, if you exercise and you increase the mass of the muscle fibers, then there will be a corresponding increase in the number of nuclei in order to keep up with protein synthesis. And this also could work in the opposite direction. For example, if you are wasting away and the muscles are getting smaller and smaller, then the number of muscle fibers will probably not change, but the muscle fibers themselves will get thinner and weaker, and you'll have a corresponding reduction in the number of nuclei per muscle fiber. Motor neurons extend outward from the spinal cord through the spinal nerves. They innervate individual muscle fibers. And we talked about the fact that a single motor neuron will innervate multiple muscle fibers within a muscle. And that unit is called a motor unit, and they'll act simultaneously to generate a certain amount of force when you have that simultaneous activation of a certain number of muscle fibers. The site where the motor neuron and the muscle meet is called the neuromuscular junction. And there are very specific, as we mentioned, there are very specific anatomical features of that neuromuscular junction. The neurotransmitter is acetylcholine. It stimulates the muscle fiber to depolarize. So normally the muscle fiber has what's called a negative resting membrane potential. And when it's excited, that voltage across the membrane of the muscle fiber changes and it depolarizes, meaning it becomes less negative. And this depolarization acts as a signal to start the contractile process. So we'll talk about this more in detail later, but the first process, which is an electrical event, is called excitation. And the second process that occurs with a slight delay is called contraction. And so we'll talk about excitation and contraction and how they're coupled. Now that we have an understanding of some of the anatomy, let's explore how muscles actually work, how they contract. The major hypothesis is concerning muscle contraction is called the sliding filament model. The basic idea is that muscle shortening occurs due to the movement of actin filaments over myosin filaments. So if you look at this diagram, we have a sarcomere. Remember, the sarcomere is bounded by a Z line at either end. The sarcomere is essentially the unit within a myofibril that allows for contraction to occur. Recall that along the midline here, there's actually protein, which isn't shown. That's called the M line. So the M line stands for the very middle of a sarcomere. And attached to the M line, you have myosin, which is a thick filament. And the myosin actually, each of these here contains, the molecules here contains maybe a certain number, a few hundred myosin proteins, and they're all stuck together. Each one's actually sort of shaped a bit like a golf club that has a long tail and then a head. And so what you're seeing here is many myosin molecules organized together to make a larger protein. And then you have the Z line. Again, those are going to be proteins. They demarcate the end of a sarcomere. And attached to these Z lines, which are actually discs in reality in three-dimensional representation, you see these as Z discs. So sometimes they're referred to as Z discs. You have actin, and these are thin filaments. And notice that the myosin filaments and the actin are parallel, but they're alongside each other. They don't end up crashing into each other. So here, shown in one, is a relaxed muscle. And so the sarcomere is long. And then you can see when the sarcomere contracts, there's a decrease in length. And there's greater overlap between the myosin and actin filaments. And you can actually see in this diagram also, there's what's called cross-bridging. In other words, the myosin heads are able to bind to the actin at specific binding sites. And so they're forming what are called cross bridges. And we'll talk more about that. So then contraction essentially can occur all the way until about what's shown here, which is the actin filaments will eventually end up more or less hitting the M line, which again is not shown here. And then you have the maximal contraction. And so we're going to look at all of this in a little more detail, but this is the overview. Basically, the idea is that the actin and myosin filaments will slide past each other. And what you'll see is that the active motor will be the myosin. The myosin heads have enzymatic activity. In other words, they can break down ATP. So they're ATPases. They can break ATP down to adenosine diphosphate and inorganic phosphate. And in the process, energy is released. So these are little molecular motors, and they're actually going to pull on the actin. And so they'll pull the Z lines on both sides towards the M line. And that leads to this shortening, which you can see here. So for example, here it shows that a sarcomere that's fully contracted. And it's actually physically limited from contracting anymore by the fact that the actin will run into the M line. I put together this diagram just to give a better idea of how this actually works. So the myosin filaments are going to pull on the actin filaments, and they'll shorten the sarcomere. So here's our picture. We have the M line, we have the two Z lines, we have the myosin, and remember so the myosin, each myosin molecule is actually golf club shaped. There's a tail and then a head. The head has ATPase activity. That's the molecular motor. These are going to reach out and make connections with the actin, forming cross bridges. So it's as if they're grabbing onto the actin, and they'll pull the Z lines towards the M line. So this Z line will be pulled in towards the M line in this direction, and this Z line on the other side will be pulled in. And so I made a diagram illustrating this with kids. Imagine you have these kids in the middle, and their object is to pull on the rope, which would be like the actin, and in doing so, they'll pull this child in the blue shirt inwards towards them, and so you get a shortening then of essentially what would be the muscle. These kids would be representing the sarcomere. And imagine, of course, you have many thousands of these sarcomeres within a myofibril and many, many myofibrils within a muscle fiber, which is a single muscle cell. And so if all these sarcomeres reduce their length by, say, 40%, then that would mean the total length of the muscle would also be reduced by a corresponding amount by about 40%. Okay, in order for the actin and myosin to interact, there has to be a signal, and the signal is calcium. Recall that calcium is at a very low concentration in the intracellular space, in other words, in the cytosol. Now, calcium is at a higher concentration in the interstitial fluid. That's the fluid outside of the cell, the extracellular fluid. And calcium is actually at a higher concentration, much higher concentration within the sarcoplasmic reticulum, which is the same thing as the endoplasmic reticulum in most cells. So calcium released into the cytosol will allow myosin to bind actin, and then allowing for these cross bridges, which I mentioned earlier. So when the muscle is in a relaxed state, this is what an actin filament looks like. There are these. So actin is actually a polymer of these globular proteins of actin, which polymerize to form a strand. It's like a strand of pearls. And there's actually two of these, and together they form sort of a helical structure where they're intertwined. Now, the thin filaments have more than just actin. There's also another protein called tropomyosin, and that lies along the actin, as shown in this diagram. And then there's a third protein called troponin. Troponin actually has 3 subunits, but we won't get into that level of detail here. Now, in the relaxed state, the tropomyosin will cover binding sites, which are shown in this diagram over here. These binding sites are places where the myosin head, which has ATPase activity, can bind to the actin. So normally these sites are covered by the tropomyosin. Now, if there's an increase in calcium within the cytosol, the calcium can bind to the troponin. Again, it has 3 subunits, so one of the subunits of troponin can actually bind to calcium. And then another of the 3 subunits of troponin is actually bound to the tropomyosin. And so when calcium binds to troponin, it then leads to a conformation or change or shape change in the troponin. And this actually pulls physically on the tropomyosin, which allows it, which now exposes the binding sites on the actin for the myosin head. And now cross-bridge formation can occur. Okay, so binding of calcium to troponin causes a conformational change that causes a dislocation of the tropomyosin, uncovering the binding sites on actin for myosin. Okay, we'll next look at how the actual process of contraction occurs. So as I mentioned earlier, in a thick filament, you have these tails of the myosin protein, and then you have the myosin head. Now, the myosin head actually has ATPase activity. In other words, it can take ATP and hydrolyze it to ADP and inorganic phosphate. And in doing so, essentially that process puts energy into the myosin head. So it's cocked, or it's similar. You can think about like putting energy into, say, a bow, like a bow and arrow, as you pull the bow back. And so we'll start off in that state here in the beginning of this diagram. You can see that this is relatively straight, and so that this is the conformation of the myosin head, in which it essentially has a kind of potential energy. And ATP has actually already been hydrolyzed, and the ADP and inorganic phosphate stay attached. Now, at least initially. And then what happens in this state, the myosin head can then form a cross bridge with the actin. Given that there's calcium, the calcium is bound to troponin. Troponin has undergone a conformational change, which then it then sort of dislocates the tropomyosin, and that exposes the myosin binding sites on the actin, which you can see here. Okay, so this is essentially ready to go. It's as if you pulled back on a bow and arrow, and you're ready to shoot the arrow. So the next step is, and there's actually an order to this. First, the inorganic phosphate comes off, and then this myosin head undergoes what's called a power stroke. So it's attached here, and now it bends. So it's essentially going back. It's from this cock state to now a relaxed state. And in doing so, it pulls on the actin filament. And so you have to imagine there's more to this protein here. So remember, there's an M line here. So the myosin continues, this protein continues this direction until it hits the M line, which is the midline of a sarcomere. And so what it's doing is it pulls on the actin. The actin is attached to the Z line here, which is actually three-dimensionally, it's a disc. So this pulls this inwards. And so this helps to shorten the sarcomere by a small percentage. Now, of course, this process would have to be repeated many times to get, say, the 40 or 50 or 60% reduction in length. So again, the inorganic phosphate comes off, and the ADP also dissociates from this. And now we'll go on to the next step. Actually, before I go on to the next step, I want to mention something just because it's a little bit interesting. So when these dissociate, the actin is moved after this power stroke, but the myosin is still very tightly bound to the actin. So potentially, this is an interesting state in which these two molecules are bound together. And if you don't have ATP available, then this is actually could be what's referred to as a rigor state, in which you could have, this is essentially a condition in which these two molecules are stuck together, and they become very rigid. And so, for example, after death, during rigor mortis, there isn't ATP available within the muscle, and so you have a state in which these two molecules are bound together, and the muscle becomes extremely stiff, and this leads to stiffness, which is characteristic of rigor mortis after death. Okay, normally, under normal conditions, ATP is available. There's free ATP within the cytosol of muscle, and so that rigor state is very short-lived. Immediately after the inorganic phosphate and adenosine diphosphate dissociate from the myosin head, there will be ATP available. It binds to the myosin head at a specific site. And once it does that, then that loosens the grip between the myosin and the actin binding site. So now they're just very loosely associated with one another. And then the next step is the hydrolysis of ATP to ADP and inorganic phosphate. And remember, they stay attached to the myosin head, but now the myosin head is moved from this relaxed state to a cocked state. So again, that's like pulling back on a bow and arrow. So you've put energy into the system, and that energy came from the breakdown of ATP to ADP and inorganic phosphate. And now the myosin head is also primed in a sense. It's now able to bind very strongly again to the actin, given that there's an actin binding site on the actin for myosin. And so basically that then brings us full circle, and we come back to the earlier point in which that they bind, and then you can go through the power stroke. I just wanted to show you this because it's the picture from your book. It's essentially exactly the same thing I've just talked about, but we can go from a resting state to a state in which we have ATP that binds to the myosin head, allowing it to release from the actin. As we said before, this would be a rigor state. So then this myosin head becomes cocked. The ATP is hydrolyzed, causing, the this is the relaxed state. ATP is hydrolyzed, then causing the cross bridge to become cocked. So it's in an energized state. Here's the thin filament. Cross bridge can now form. Inorganic phosphate is released, causing a conformational change. So that causes then the power stroke. And after the power stroke, the ADP can dissociate. And now we're back to the state where, again, it's called the rigor state, where they're tightly bound. And they will stay bound until there's ATP available. And then we come back here, new ATP binds to the myosin head, allowing it to release from the actin. And then we go all back through this whole, circle all over again. So in order for muscles to contract, as I mentioned, we need ATP to be available. Otherwise, the muscle might end up in a rigor state. So ATP is required for muscle contraction, as the release of energy from ATP hydrolysis provides energy for the power stroke. The myosin ATPase breaks down ATP as the fiber contracts, and you get this reaction where ATP becomes ADP and inorganic phosphate. So what are the sources of ATP? Well, we've actually talked about this. So in previous class lectures, so phosphocreatine is one source. Remember, phosphocreatine can donate a phosphate group to ADP, regenerating ATP, which can then be used for muscle contraction. So you have, within muscles, you do have a reservoir of phosphocreatine. So that's a very readily available source for phosphate to regenerate ATP from ADP. You have glycolysis, which you can recall as the breakdown of glucose, yielding 2-pyruvate, and you get a net gain of 2 ATP per glucose molecule. And then, of course, we've talked about oxidative phosphorylation. And so if you can fully oxidize glucose, you'll get a far higher yield of ATP. This is a diagram from your book showing some of the things we just talked about. So glucose can enter into a muscle fiber from the blood. You can go through glycolysis to produce ATP. Glycogen is found within muscle itself, and it can go through glycolysis to form ATP. If there's no oxygen available, the pyruvate, which is formed by glycolysis, may be converted to lactic acid. Now, the other possibility is if there is oxygen available, you can go into oxidative phosphorylation. And this yields a much higher amount of ATP. Now, fatty acids, there may be free, we talked about free fatty acids. There are situations such as, long-term exercise, like, if you're exercising for, say, over 90 minutes, in which you may have lipolysis. This leads to an increase in free fatty acid concentration in the blood. These fatty acids can be oxidized, again, via oxidative phosphorylation, leading to ATP. This is a little more unusual, but there are situations such as starvation, perhaps very strenuous exercise for long periods of time, in which your body may actually start to break down protein into amino acids and then use those amino acids for oxidative phosphorylation. Now, the ATP may be used for a couple different things with regard to muscle contraction. One is that the myosin itself has an ATPase activity on the myosin head, and that helps power the contraction. So it can be thought of as a molecular motor. Now, we talked about calcium as being a signal that leads to contraction of the muscle. So calcium is normally at a very low concentration in the cytosol. So in order for the muscle to relax, the calcium has to be pumped back into the sarcoplasmic reticulum. It's pumped uphill, in other words, up its concentration gradient. So that's active transport, meaning that it takes energy in the form of ATP. So this pump also takes quite a bit of energy, and so you would need any ATP to run that pump. One term you may hear a lot is excitation-contraction coupling. By excitation, we're referring to the electrical activity in, first of all, the motor neuron, but then there's also electrical activity in the muscle itself. So the sequence of events is such that initially an action potential, which is an impulse or spike, which is a change in electrical potential across the membrane of the axon. So normally the axon's at negative 70 millivolts roughly. During an action potential, it transiently increases past 0 to about positive 30 millivolts before returning. So this electrical event can actually propagate or travel down an axon all the way to the axon terminal at the neuromuscular junction. Neurotransmitters released, acetylcholine is released into the synaptic cleft, and then there's an end plate on the muscle, which consists of a specialized membrane that has a number of receptors for acetylcholine. These receptors are ligand-gated, meaning the acetylcholine will bind to them, and then the channels will open, and they'll allow positive ions to flux into the muscle. Now, normally the muscle has what's called a resting membrane potential, meaning it has a negative potential within it relative to the voltage outside of it. Positive ions moving into the cell will make the cell depolarize. And so you get a small depolarization in the region of this neuromuscular junction. Now, typically this depolarization is great enough that it'll actually trigger what's called a spike. And I've drawn a spike here. This is starting at negative 70 millivolts in the muscle. Then something like this occurs and it triggers an action potential. So this is a change now in membrane potential. The voltage of the cell will go up to about plus 30 millivolts before dropping actually below 70 and then returning to the regular resting membrane potential. So this electrical event is called a spike or action potential. Sometimes it's called a nerve impulse. And it'll actually travel in both directions along the muscle fiber. So this electrical event is what was referred to as excitation. And this leads to contraction. So with a bit of a delay, you can see in this picture here, we have an action potential. Time is on the x-axis. And with a bit of a delay, there's an increase in tension within the muscle fiber. And this increase in tension is called the twitch. So a single action potential leads to a single twitch. So essentially we have the excitation, a slight delay, and then the actual contraction within the muscle fiber. So again, we have depolarization at the motor end plate. That's the specialized membrane in the muscle cell just below the neuromuscular junction, which is essentially a synapse. So this depolarization leads to excitation of the muscle, which then generates an action potential. And then that then leads to the muscular contraction. So in order to effectively lead to muscular contraction, the action potential travels not only to the two ends of the muscle fiber, but also down those continuations of the membrane called transverse tubules that actually go all the way through the muscle itself. So those tubules essentially bring this wave of depolarization or excitation into the muscle cell. And then there are these special channels called voltage-gated calcium channels that open. And due to this trigger, they then release calcium from the sarcoplasmic reticulum. Recall that anatomically, the transverse tubule is very close to the terminal cisternae, which are parts of the sarcoplasmic reticulum. So those are physically juxtaposed. They're very close. And that allows for this coupling here. So calcium then is released from the sarcoplasmic reticulum within the cells, within the muscle fiber. The calcium then binds to troponin, causing a position change in tropomyosin, and that exposes the myosin binding sites in the actin. So we've talked about that previously. And then we can have a strong binding state formed between the actin and myosin, and that allows it to form a cross bridge. And then once that cross bridge is formed, immediately after that, you can go into the power stroke, which we talked about before, and that will actually pull the myosin head will actually pull on the actin, thereby bringing the Z line closer to the M line. And that, repeated many times with many of these myosin heads pulling on different actin filaments and so forth, will lead to actual contraction of the muscle. So I'd like to talk a little bit about the excitation phase here. So again, the nerve signal arrives. This is the action potential, arrives at the synaptic knob, also called the synaptic bouton. Bouton means button in French, or axon terminal. So it arrives there. And then acetylcholine, which is a neurotransmitter, is released into the synaptic cleft, and it diffuses across and it binds to receptors on the motor end plate, which is that specialized postsynaptic membrane on the muscle cell. Now, these receptors are ligand-gated, meaning they're triggered by a chemical such as acetylcholine. In this case, they're specific for acetylcholine. They're also ion channels. And so when acetylcholine binds to the receptors, these ion channels open. And sodium, you may recall, is in a higher concentration outside of a cell compared to inside. And so sodium now will move into the cell, into the muscle fiber, causing a depolarization. That depolarization is strong enough that it will it'll trigger or cause an action potential, which is an even greater depolarization event. And this actually is conducted or propagates from the site of the neuromuscular junction, which is typically sort of midway at the midpoint along the length of a muscle fiber. And that action potential will actually be generated in both directions and go to the very ends of each the muscle fiber. At the same time, the action potential is conducted downwards and into the muscle fiber itself via the transverse tubules, which go all the way through the muscle fiber. Again, I realize I've talked about this, but just to make this clear, so contraction then occurs when you have the depolarization of the T tubules. So the action potential travels down the T tubules, causing release of calcium from the sarcoplasmic reticulum, specifically within those terminal cisternae, which are located very close to the transverse tubule. Calcium binds to troponin, causing a shift in tropomyosin, which uncovers the myosin binding site on actin. The myosin binds to actin, forming a cross bridge. Inorganic phosphate is released from the myosin. And then you have the actual power stroke, which is this cross bridge movement. Now, at that point, recall that the myosin is now stuck very tightly to the actin, and that's called a rigor state. And if you don't have any ATP within the muscle, then that would actually lead to rigor mortis, in which the muscle becomes very stiff. But normally you do have plenty of ATP. There's a certain concentration of ATP within your muscle cells at all times. This immediately attaches to the myosin, breaking that cross bridge. The ATP is hydrolyzed to ADP and inorganic phosphate, which energizes the myosin, and that's referring to this cocking of the myosin, where it changes its conformational change. It sort of stretches out, and that's again equivalent to sort of drawing back on a bow and arrow, and putting energy into that myosin protein so that it can then do another power stroke. Okay, so we've talked about how contraction occurs and how through these many little cross bridges forming and unforming and unbinding and so forth. And you have to remember this is sort of happening in asynchronous. It's not as if all the myosin cross bridges are sort of coordinated to pull at the same moment. There are hundreds of them, and they're doing asynchronously, and of course we've just talked about one myofibril, but there are many myofibrils, which are these long structures that are involved in contraction within each muscle cell. So there's many thousands and thousands of these occurring. And if they're all doing this cross bridge formation, power stroke, and so forth, that's going to lead to contraction. But you wouldn't want your muscles to be contracted all the time. They have to also relax. So let's talk about how that occurs. So eventually what happens, the motor neuron stimulation ends, meaning it stops sending action potentials down the axon. At this point, acetylcholine is no longer released. And the acetylcholine that's in the synaptic cleft is actually enzymatically degraded. And so there's a reduction in the concentration of the acetylcholine in the synaptic cleft. It doesn't bind to those postsynaptic receptors in the end plate of the muscle fiber. And so you don't have the depolarization occurring anymore. There's no more stimulus. So the muscle fiber then repolarizes, meaning it goes back to its regulated negative resting potential, which is about negative 70 millivolts. And that's just sort of a standard resting rate. In fact, it's probably muscle fiber is a little bit more negative than negative 70. It might be more like negative 90 or so. I mean, yeah, maybe negative 80. But in any case, that's this normal resting state when it's not being stimulated. Okay, so that excitation first goes away. Now it's calm electrically. Now the contraction is still going on, but what happens is calcium in the cytosol is always being pumped back into the sarcoplasmic reticulum. And so as soon as you stop the excitation, the calcium pumps essentially catch up and they keep pumping the calcium back. And so they lower the concentration of calcium within the cytosol of the cell. And so then at that point, the troponin, which the calcium unbinds for the troponin. Troponin has a conformational change, and it goes back to a situation in which it pulls in the tropomyosin, and now the tropomyosin returns to its original position, which is covering the myosin binding sites on the actin. Now the myosin can't form cross bridges with the actin, and the muscle relaxes as a consequence. Okay, here's a few take-home messages. The muscular contraction is explained by the sliding filament model, which proposes that muscle shortening occurs due to the movement of the actin filament over the myosin filament. Excitation-contraction coupling refers to a sequence of events in which the nerve impulse, the action potential, depolarizes the muscle fiber, and leading to, with some delay, a muscle shortening by cross-bridge cycling, which we've just talked about. The trigger to initiate muscle contraction is the depolarization-induced release of calcium from the sarcoplasmic reticulum. And again, we looked at that with a little more anatomical detail. It was specifically associated with those transverse tubules are the terminal cisternae. So it's most of the release is occurring in that region of the sarcoplasmic reticulum, and that the calcium then ends up in the cytosol. And then that leads to the contraction, the cross-bridge cycling, due to cross-bridge cycling. So muscular contraction occurs via the binding of the myosin cross-bridge to actin and the repeating cycling of myosin pulling on the actin molecule, resulting in shortening of the muscle fiber. Muscle relaxation occurs when the motor neuron stops exciting the muscle fiber. And calcium, of course, is continuously being pumped back into the sarcoplasmic reticulum. So eventually those pumps catch up and they reduce the cytosol calcium concentration such that relaxation can occur. This removal of calcium from the cytosol causes a conformational change in troponin, which in turn causes a position change in tropomyosin, which blocks the myosin cross-bridge binding site on the actin molecule, resulting in muscle relaxation. So one interesting topic is muscle fatigue. There are many different theories about muscle fatigue, and typically you might see a very sort of simple solution. Like people say, well, it's just the accumulation of lactate. Turns out that's not really true. There are many muscle fatigues. One of the most complex phenomena you could possibly study. First of all, there is a central component to muscle fatigue. So that occurs within the central nervous system. So if you're exercising, and this is occurring at a subconscious level, it's not that you're being lazy or something like that. But the central nervous system is very good at detecting that, the fact that you should be fatigued because of the level of exertion you've been doing. And so there's a phenomenon of central fatigue, which we're not going to really go into much here, but that is a phenomenon that does significantly contribute to muscle fatigue. Now, there are other reasons for muscle fatigue that occur within the muscle fibers themselves. So first of all, you'll see a decline in muscle power output. So this is essentially a definition of what fatigue is. And this power output decline can be evident both as a decrease in force generation as well as a decrease in shortening velocity. And the reasons for this actually vary depending on the type of exercise. So for example, if you did a short sprint for 60 seconds at a very high intensity exercise, well, then you will be doing anaerobic metabolism, so you would expect an accumulation of lactate. So that does, in fact, have some influence on causing muscle fatigue because it interferes with cross-bridge cycling, which we've talked about. At the same time, lactic acid that formed, is able to donate a proton to the solution, and so it can change the pH of the solution. Again, that has a negative effect on cross-bridge cycling. We already mentioned the fact that in order for cross-bridge cycling to occur quickly, you need to have a relatively high concentration of ATP. So if you start to get to the point where you actually have a little bit of a reduction in ATP, that may affect cross-bridge cycling slightly. I mean, it might decrease the rate at which it can occur. And then, of course, there's also the possibility of these things called free radicals also being generated, and those can damage tissue and so forth. So all of these different phenomena can diminish the cross-bridge cycling between myosin and actin. Okay, for longer duration exercise, like say a more lower intensity but longer exercise, and we're talking about 2 to 4 hours, there's a number of other muscle factors that could contribute to fatigue. One is the accumulation of free radicals. Now, normally, a small amount of free radicals is expected. It's not necessarily that damaging, but because you do have mechanisms to deal with them. But an accumulation of free radicals could potentially interfere with things like cross-bridge cycling. With more longer-term exercise, you may have changes in electrolytes within the blood and therefore within the interstitial fluid. By electrolytes, we're talking about things like sodium, potassium, chloride, magnesium, calcium, and so forth. So any sort of imbalance in those could lead to a certain level of fatigue within muscles. You would never get this with a very short-term intense exercise. It's very unlikely you'd have electrolyte imbalance. But with a longer duration exercise, it's possible, for example, for a person to become dehydrated. These imbalances can occur. Finally, you can also have glycogen depletion. So in your body, you have the majority of your glycogen is in skeletal muscle. There's also some in the liver. Both those sources of glycogen, which are a good source of carbohydrate for glucose, both of those can get depleted because you simply don't have that much of it. And so if you were just resting, for example, just to give you a sense of scale, the liver has enough glycogen to keep you fueled for about 24 hours. But that's just in a resting state. So you can imagine the glycogen could start to become depleted within a shorter period of time if you're exercising. Now, it never really becomes depleted because there are other mechanisms that maintain the glycogen, say, within the liver, such as gluconeogenesis. But also within at the level of the skeletal muscle itself, you can start to see glycogen depletion. And so that would potentially lead to some fatigue. So this is just a simple diagram of what muscular fatigue would look like. If you were looking at force production, at the onset of fatigue, you might see a decrease in force production. And at the same time, contraction time would also be affected. So contraction time would increase. And so both of those would be indicators of muscular fatigue. So our take-home messages are that muscular fatigue is defined as a reduction in muscle power output, and you may have decreased muscle force generation, as well as a lower shortening velocity. The causes for exercise-induced muscle fatigue are very complex and very depending on the type of exercise performed. So we just saw that a second ago. Depending on short-term, very short, high-intense exercise will lead to certain phenomena occurring, and with more long-term, lower-intensity exercise, other phenomena will occur that lead to muscle fatigue. And again, just to remind you that muscle fatigue isn't just in the muscle. There's also a very strong CNS or central nervous system component to muscle fatigue. And again, that's not something sort of psychological or anything. It's not like a person's giving up or being lazy or something. That's just a built-in part of how your brain interacts with your body, and that's called central fatigue.